Good morning, friends. Myself Reshma Swaraj. I am currently pursuing post graduation in Department of Biotechnology in Sri Krishnadevaraya University at Anantapur. Today, I would like to discuss about DNA replication. Let us go to the contents. Here, there is an introduction, basic rules for DNA replication, enzymes involved in DNA replication, process and role of enzymes in DNA replication. Let us go to the introduction. In molecular biology, DNA replication is the biological process of producing two identical replicas of DNA from original DNA molecule. DNA replication was postulated by Watson and Crick after they discovered the structure of DNA. DNA replication is a semi-conservative. Each strand in double helix acts as a template for synthesis of a new complementary. The process of DNA replication is vital for cell growth, repair, and reproduction in organisms. Here, there is a sentence DNA replication is a semi conservative. What is meant by semi conservative? Let us know that. Here, there is a parental strand which is act as a template that produces two daughter molecules. One is made up of parental strand and another is newly synthesized. That is known as semi conservative. Next, the basic rules for DNA replication. DNA replication is always a semi-conservative. DNA replication begins at sequence called origin. DNA synthesis is initiated by short fragments of RNA called primer. The elongation of DNA strand is always in the 5' to 3' direction. DNA replication can be uni or bidirectional. Replication is continuous on leading strand and discontinuous on lagging strand. New nucleotide strands are complementary and anti-parallel to their template strands. DNA replication takes place at very high rates and astonishingly accurate due to the process of nucleotide selection, proofreading, repair mechanism. Next, what are the enzymes and proteins involved in DNA replication are DNA helicase, DNA primer, RNA primer, single stranded binding proteins, DNA polymerase, DNA gyrase, DNA ligase. Now, let us know the process of DNA replication. First, replication fork formation. Before DNA can be replicated, the double stranded molecule must be unzipped into two single strands. DNA has four bases called adenine, thymine, cytosine, and guanine that form pairs between the two strands. In order to unwind the DNA, these interactions between base pairs must be broken. This is performed by an enzyme known as helicase. The DNA helicase separates the strands into Y shape known as replication fork. Here you can observe that this helicase binds to the double stranded DNA and forms the replication fork and also separates the double stranded into a single stranded molecule. And next, DNA gyrase. As it unwinds, it causes the DNA to supercoil, which increases the stress of the DNA and makes it difficult to unwind. A special enzyme known as DNA gyrase is also known as topoisomerase to remove this stress and prevent the supercoil of DNA. Here you can observe that the topoisomerase binds the double-stranded DNA and prevent the supercoiling of that double-stranded DNA. Next, single-stranded binding proteins. During DNA replication, single-stranded binding proteins binds to newly separated individual DNA strand, keeping the strands separated by holding them in place so that each strand can serve as a template for new DNA synthesis. And it also helps to prevent the reassociating of hydrogen bonds present between the two strands. Here you can observe that the single-stranded binding proteins binds to the single-stranded DNA and prevent the reassociation of the two strands. Next, primer binding. Once the DNA strand has been separated, a short piece of DNA called a primer binds to the 3' end of the strand. The primer always binds at the starting point for replication. Primers are generated by the enzyme DNA primase, the primer which helps to initiate the synthesis of a new strand. Here you can observe that RNA primer is attached and initiates the formation of a new strand. It is the initiating primer to synthesize the new strand. Next, DNA polymerase. Enzyme known as DNA polymerase are responsible for creating new strands by the process called elongation. In this, DNA polymerase binds and forms the complementary strands to the template strand from 3' to 5' direction. The leading and lagging strands are formed. Here, what is meant by leading strand and what is meant by lagging strand? Let us know that. The leading strand is a single DNA strand. During DNA replication, it synthesizes 3' to 5' direction. It synthesizes continuously by adding DNA in same direction. It requires only one primer. The new strand is known as leading strand. Here you can observe that the leading strand is a same direction as replication fork and it is continuous in form by adding the DNA and form continuous leading strand. And next one is lagging strand. The lagging strand during DNA replication which synthesizes 5' to 3' direction, it synthesizes discontinuous manner. It forms opposite to the replication fork. It requires more primers. It synthesizes like small fragments. That fragments are called Okazaki fragments. The new strand is called as a lagging strand. Here you can observe that the lagging strand is in opposite direction of that replication fork and it is in a discontinuous manner and it looks like say fragments and that fragments are called the Okazaki fragments and this strand is known as a lagging strand. Next one is Okazaki fragments. What are the Okazaki fragments? Okazaki fragments are short, newly synthesized DNA fragments that are formed on the logging template strand during DNA replication. After the synthesis of both leading strand and lagging strand, the Okazaki fragments are joined together by a specific enzyme called as DNA ligase. This DNA ligase removes the gaps between them and forms a continuous strand. Then finally two newly synthesized strands are formed. Whereas coming to the conclusion, the purpose of DNA replication is to produce two identical copies of DNA molecule. This is essential for cell division during growth or repair of damaged tissues. DNA replication ensures that each new cell receives its own copy if the DNA. In that two identical copies, one is parental strand and other is newly synthesized one. Totally, the DNA replication is done by some enzymes and also forms the newly synthesized strands and form a one parental strand and one newly synthesized one. Thank you.